Yeah. Hello, everyone. Thank you for joining us today. My name is György Miklós. I'm VP of Sales and Marketing in Enamine. And today we're going to talk about fragments and the recent advances in covalent modifiers. But before before I introduce our current speaker and basically you start listening to the talk, I would like to just remind you, a few rules for the webinar. First of all, you will be muted during the whole course of the webinar. And if you have any questions and would like to ask our today's speaker, please use this Q&A chat box, which is located at the bottom of the screen. Once webinar is finished, you will receive the link to the Zoom Cloud storage with the webinar, so you can still have access for it to approximately for 1 week or so. And also once we have finished the webinar, it will be available on our page on YouTube. Yeah, so please follow, and as by the way, you can find other interesting webinars there as well. So yeah. Today, again, as I said, today we discuss recent advances in covalent fragments, and it's a really great pleasure for me to introduce to you Dr. György Keserű. You probably know him for his works in that field. Just again, quick recap who he is. So he basically graduated from Budapest University of Technology and Economics, that's in Hungary. He's been working in Sanofi Avances, in Gedeon Richter, and currently he works as a principal investigator at the Research Center of Natural Sciences. So with that, please listen to our today's presenter. György, the stage is yours.
Okay. So thank you, György, for the kind introduction, and also I would like to welcome everybody who joined to this webinar. Just let me start by sharing the screen. I hope you could see the screen here. Just György, would you please?
Yes, we can see. Yes, we can see the screen. Yep.
Okay. So again, first of all, I would like to thank the organizers and all the Enamine staff inviting me to be participated to the webinar. And the next little bit more than half an hour, I would like to speak about the identification of different binding sites using electrophilic MiniFrags we developed in the recent years. So when we started to go with covalent fragments, we considered the advantages of fragment-based drug discovery and covalent mechanism of action. We know that in the case of fragment-based approaches, we have an option for the enhanced sampling of the chemical space, and we have higher chance of binding for the fragments that typically much less complex as compared to larger molecules. They show typically better physical chemical profile, and they provide rational optimization strategies, especially in the case if you have structural information on the binding of these fragments. On the other hand, there are some certain advantages associated with the covalent mechanism of action, and these are typically the enhanced potency, the higher selectivity, better pharmacodynamics, and of course, the prolonged duration of action that is directly connected to the formation of the covalent bond to the target site. So if we summarize these kind of advantages, then covalent fragments would provide probably easier detection, enhanced potency, improved target engagement, and at last but not at least, fixed binding mode for optimization that probably makes their optimization a little bit easier as compared to the loosely binding, weak, non-covalent fragments. When we considered that what kind of covalent fragments would be exciting, then we analyzed a little bit the warheads actually available for covalent modification of different targets. And you could see on the right-hand side of the slide on the warhead distribution that many of the compounds already reported as covalent compounds, they have acrylamides or other Michael type addition or Michael type acceptors for the covalent modification. There are, however, some other options, some other chemistries available for covalent modification, and that includes non-Michael type addition reaction, addition eliminations, nucleophilic substitution reactions, and oxidation. But clearly, it seems that basically the 2/3 of the covalent approaches are uses the very common, very popular Michael addition. So what we would like to do that we analyzed the composition of the Michael acceptors. I mean that typically if they are, if a Michael acceptor pod is installed on a non-covalent ligand, that needs at least 3, but most often more than 4 heavy atoms. And if you consider the binding mode of the original non-covalent ligand, if you add 3, 4, or even more heavy atoms to the non-covalent scaffold, this would affect the molecular recognition and the binding mode of the whole molecule. So therefore, we thought that what about if we are trying to minimize the extension of the original non-covalent core to 1 or 2 atoms, and this is how we reached electrophilic heterocycles. So we started with the relevant heterocyclic cores used very often in medicinal chemistry, and we installed activating warheads that could react typically in the nucleophilic, aromatic nucleophilic substitution reaction or nucleophilic additions due to the electron redrawing character of the heterocycles. And as I already mentioned, that in this approach, we were able to minimize the extension of the original non-covalent heterocyclic core, because in this approach, we are using maximum 1 or 2 atoms. So what we did, we derived different 5-member, 6-member heterocycles with suitable substituents up to 1 or 2 atoms. And we synthesized these compounds, and first we profiled their reactivity in a surrogate assay. And then we turned to biophysical and biochemical screening. We performed these screening for some different therapeutically relevant targets. And our basic objective was just to generate relevant covalent starting points for further optimization. So let me show you a little bit about the chemistry. So on the left-hand side of the slide, you see a different nitrogen-containing heterocycles, both the 5-membered and the 6-membered heterocycles that contains 1 or more heteroatoms, and substituents indicated by the R groups that includes the halogens, the nitrile, the vinyl, and the ethinyl group. In addition to these reactive electrophilic heterocycles, we also created their N-methylated analogs, just improving the electrophilicity of these fragments. And we derived these N-methylated derivatives by simply methylation reaction, typically performed at acceptable or even high yield. Using this approach, we were able to cover quite a large range of reactivity as measured in the GSH half-life. Why we think that this approach would be interesting? Because probably you know, many of you who are already involved in fragment-based drug discovery programs knows that Astex previously introduced a new approach that is called MiniFrag approach. So typically, Astex is using MiniFrags that are pretty much similar to our set of electrophilic heterocycles, but they are not reactive at all. So they are using these small, very small, ultra-low molecular weight compounds for X-ray screening. And they demonstrated that screening these very, very small fragments, MiniFrags, this is useful to identify not only the binding hotspot, but also the warm spots as well that are located close to the hotspot. And therefore, these warm spots could be used for identification of the growth vector, and it would provide a more straightforward optimization of the fragment bound to the original hotspot. On the other hand, it is pretty much important that just because of the ultra-low molecular weight of these compounds, they provide enhanced sampling of the chemical space, and they also demonstrated that these very small fragments are able to detect unprecedented ligand binding packets on the surface of the protein target. So we think that most of these advantages already demonstrated for the non-covalent version of the original MiniFrags will be valid for the covalent version as well. So let me show you first the characterization of our covalent MiniFrags library against GSH as a thiol surrogate. In these tables, on the top row, you could see all of the heterocyclic cores we considered. And these blue spots are indicating the exact position of the substituents. And the different substituents you could see in the subsequent rows here, the chlorine, bromine, iodine, cyanide, vinyl, and atenyl group. A pretty much similar table is available for the N-methylated analogs here. And those fragments that showed some significant reactivity against GSH, these are colored green. And you could see that we were able to identify reactive fragments in both the non-methylated and the methylated versions of these small heterocycles. The good news was that there was no stability issues detected in buffer conditions when we tested the fragments against GSH. However, on the other hand, these large set of fragments, altogether 145 small fragments, was able to cover the diverse range of reactivity. And finally, I would like to mention that these small heterocyclic fragments provide us an opportunity to tune the reactivity of the warhead, and by introducing further substituents, and therefore we can prioritize residue-specific warheads from this set of compounds. So having the library characterized against the surrogate, the next point was to test this library against different proteins of interest. And for this purpose, we used biochemical and biophysical methodologies. In the case of biochemical screening, we typically detected the residual activity of the protein, and we measured the time-dependent IC50s. Also, we try to identify viable chemical starting points for these particular targets. But of course, just because of the biochemical detection, typically we have no information on the mode of action. In contrast, if we are using biophysical methods for the detection of covalent binding, then both mass spectrometry, NMR, and X-ray, I will show you how these methodologies would provide information on the binding side of our fragments, and that would facilitate their optimization to a full targeted covalent inhibitor. So typically what our strategy was that we first tested the full library against the protein of interest using biochemical methodologies, just confirming the functional relevance of the binding site labeled by the covalent fragment. And we used biophysical techniques for secondary screening, just validating the hits and also to identify the exact site of labeling. So here we have a pretty large number of different proteins we tested the library. This includes the classic enzymes, yeah, like murate, MO, ANB, thrombin, PBP1B, factor 10A, the acetylcholinesterase, butyrylcholinesterase, and some benchmark proteins like lysozyme, myoglobin, and carbonic anhydrase. We also tested the library against more challenging compounds, including the GTPase KRAS G12C, against GPCRs, epigenetic targets like HDAC8 and other HDACs, and the COVID target of SARS-CoV-2 main protease. In this talk, I would like to focus to the KRAS, the HDAC8, and the 3CL-Pro efforts. Just let me show you first library profiling, and I would like to ask you just first focus on the right-hand side of the slide, and here you could see the different targets, including the GSH surrogate as well, and also the large amount of different fragments listed in the different rows, and you see different tones of blue colors that indicates the activity of different fragments against different protein targets. So you could see here that KRAS G12C having one very important and incriminated cysteine residue showed typically higher reactivity as compared to the other targets. However, in the case of MOs and also murase that having also some characteristic cysteine residues, even the non-methylated, less reactive MiniFrags were able to label these targets. If we now are going to the left-hand side of the slide, this is library profiling for the N-methylated version. And here you could see a much larger number of highly reactive small fragments, and that is due to the methylation of the different fragments. And therefore, we use this part of the library as well, just covering from the less reactive to the more reactive compounds. I would like to focus first on the KRAS G12C example, and here I show you just starting in an ARS-like acrylamide, indicated on the left-hand side, we replaced the original Michael acceptor acrylamide to a heterocyclic version that contains a vinyl group that is basically a masked version of the Michael acceptor here. And we investigated first the binding site of this compound, and as you could see here, that we identify the most important interacting amino acid residues using HSQC NMR technology. And on the basis of these investigations, we confirmed that the binding site of both of the covalent KRAS G12C inhibitor is basically the same. At the bottom, you could see the binding mode of the heterocyclic version of the G12C inhibitor. And here you could see the biological activity on different cell lines, including LCLC 103H that expresses the wild type KRAS, and the H1792 that expresses the G12C mutant. And as compared to the original version, that we observed more or less the same IC50 and the selectivity as it was found for the original acrylamide version. Next, I would like to go to a somewhat more interesting target, and this is HDAC8. And here you could see the results of the first biochemical screening of the whole library against HDAC8. And you could see here that in this case, we observed only very few non-methylated MiniFrags indicated by green that showed some reactivity against HDAC8. In contrast, we found many of the unmethylated heterocycles that was able to label and able to inhibit HDAC8 very effectively. So this is a clear indication that a high reactivity of these small fragments are pretty much important to identify high quality starting points. And if we compare the typical or the average IC50 observed for the non-methylated and methylated MiniFrags, there is a significant difference since the IC50 for the methylated MiniFrags is much lower than that was found for the non-methylated version. So that's why we focus pretty much to the 11 best fragments. These were basically all methylated fragments. And what we did, first we tested these compounds against different cysteine mutants of HDAC8. And you could see the results here. The green values, again, are those fragments and those mutants that showed some significant labeling on the basis of the biochemical test. And these were basically confirmed by a mass spectrometry, just identifying the exact binding site on the surface of HDAC8. And the next slide, you could see that where exactly these binding sites are located. So first, I would like to focus on the known sites. These are basically labeled by cysteine 102 and 153. These cysteines are the members of the redox switch that is needed for the powerful function of HDAC8. And there are also some activity regulator cysteine residues, the cysteine 275 and 352, that are also involved and also labeled by a few unmethylated small heterocycles. More interestingly, however, we were able to identify new sites as well. And let me start with this one, this C28 rather large number of the best compounds were found to label this side, as well as the C314 that is located on the opposite side of the H-8 surface. And finally, I would like to mention this cysteine 244 that gave us altogether 6 hits. And together with the other two cysteines, cysteine 28, 244, and 314, these might be considered as potential targets for developing new allosteric covalent H-8 inhibitors. So we wanted to use at least one of the hits we identified for the design of new targeted covalent inhibitor, and we picked up a compound that was previously co-crystallized with H-8 and having an aromatic residue, a dichloro residue indicated here, that we found this dichlorphenyl moiety are pretty close to cysteine 153. And that's why our idea was that, and what about if we replace this particular dichlorophenyl moiety with one of the electrophilic heterocycle hits we identified by biochemical and biophysical screening. We selected a vinyl analog for this purpose, and we designed compounds with different connection points and different link lengths to find the best position and the best linker of the non-electrophilic core and the newly identified N-methylated electrophilic fragment. So what we did, we created these compounds here, and the first challenge was we tried to dock these compounds into the binding site of the original compound. And here you could see that what we did, we compared the orientation and the binding mode of the design compound to that of the non-covalent analog. And we identified a different compound with pretty nice docking scores. And in the first iteration, we synthesized these two compounds, both the non-methylated and the methylated analogs of this pyridine derivative. Unfortunately, however, when we investigated both of the compounds against the wild type H-8, we did not observe any binding. And probably the case was that the linker region of these compounds seems to be too large, that prevented the best fit of the design compound to the available binding site. So that's why in the second iteration, we generated compounds with much shorter linkers. And fortunately enough, we found that these two compounds, ABP 752 plus, and also the other one, even the shorter linker with ABP 759 plus, showed binding to the H-8 site we targeted. Of course, we found much better binding for the little bit longer linker. So this is basically a medium size linker as compared to the original we tried to utilize in the first iteration, and the really short one. that provided a low micromolar activity as a covalent HDAC8 inhibitor. And fortunately enough, this compound showed activity on both of the cell lines that are expressing the HDAC8 protein. And we were very happy that even using this very simple iteration and optimization process, we were able to achieve the cellular efficacy for one of the compounds we identified. The last example I would like to share with you, this is related to the 3CR-Pro, the main protease of the SARS-CoV-2 virus. And this study was done in collaboration with the Diamond Light Source X-ray facility that screened a pretty high number of different fragment libraries, including our Covalent MiniFrags against the main protease of the virus. And during this screening, they were able to identify 2 hits. Interestingly, both of the compounds have the cyano group that form covalent interactions with the active cysteine residue of this cysteine protease. And we observed a similar contact and similar formation of the covalent bond for both the 5-membered and the 6-membered ring. And starting from these 2 hits identified from the library that at that stage does not contain the N-methylated analogs, only the non-methylated ones. And next, we try to optimize these initial fragment hits using an optimization protocol I would like to show you here. So we started from the PDB structures of the 2 covalent and other 17 non-covalent, non-overlapping fragments. And we try to select those fragment pairs that are suitable for linking. We found altogether 16 of these pairs. And the next step, we used the delinker technology for the linker design, and we finally obtained 20 linked pairs that were docked to the binding site using the Schrödinger's glide covalent docking mode. And we checked the binding modes, and finally, we compared the binding modes of these linked molecules to that of the original fragments, just to realize any movement of fragment positions upon linking. Finally, we selected 3 linked pair for synthesis and in vitro testing. And in the next slide, I would like to show you that we started from the 6-membered nitrile that showed basically no activity against the main protease. And we selected this sulfonamide that is otherwise also a fragment hit identified by the XChem people. However, unfortunately, this compound also showed no activity against the main protease. So again, we used the linker design technology and synthesized compounds with 3 different linkers and 3 different connection points. And fortunately enough, we identified 1 compound with a relatively short linker showing a low micromolar activity against the protein target. And if we consider the lack of activity of the starting fragments, then this is a significant improvement in the efficacy of the linked compounds. So at the end of my talk, I would like to summarize that it was my pleasure to show you how we developed the Covalent MiniFrag library that contains altogether 145 small electrophilic heterocycles that were characterized against GSH and also other amino acid surrogates and were tested against multiple targets. So we describe here many new warhead chemotypes that provide a tunable reactivity, just introducing further substituents to the heterocyclic core. So most importantly, we were able to identify unprecedented age-decade binding packets that might be useful for covalent targeting of this therapeutically relevant and interesting target. And we also showed that small and reactive heterocycles are suitable to incorporate to known inhibitors, like in the case of the KRAS G12C, or we can use these inhibitors for fragment linking, as it was demonstrated in the case of the SARS-CoV-2 main protease inhibitors. If you are interested there are some further details available. Our JMedChem paper published earlier this year, and it is important that the Covalent MiniFrag library is available for screening at a diamond light source, and of course available for Enamine. So at the end of my talk, I would like to acknowledge all of the contributors from my institute, RCNS Hungary, but also Darmstadt University. We worked together with Francis F. Meyer-Alms on the HDAC-8 project. We are particularly grateful for the team working at a diamond light source, and thank you very much for your kind attention. So now I'm stopping sharing the screen, and I would be more than happy to answer any question or comment.
Yeah, thank you, George, for this really excellent talk. So we have a few questions here. So first of all, have you considered sulfone or sulfoxide leaving group? If not, why not?
That is a very good point. Frankly speaking, we did not consider. However, it is very reasonable to try that. So basically, when we selected the substituents of the electrophilic heterocyclic cores, we focused pretty much on the two reaction types, and we tried to exemplify both the nucleophilic substitution reaction and the nucleophilic addition reaction as the two major types of chemistries we can use for amino acid labeling. However, it is a very good point that we can now extend the functional groups that are available for both types of reactions. And that is also an option to tune further the reactivity of the small MiniFrags.
Yeah, thank you. Another question: if you considered N-oxides along with your metal compounds, and if so, how do the directivity of the former related to the directivity of the N-methyl compounds?
So basically, the idea of N-methylation came from an Angewandte paper published by Maria Matos earlier, and the Matos group demonstrated that they were able to increase the reactivity of the nitrogen heterocycles very much by N-methylation. So that's why we use this technology. It was very straightforward and provided us a pretty high number of N-methylated compounds. However, probably you guys observed that we were basically unable to N-methylate all of the heterocycles, and I guess that this N-oxide preparation would be even more challenging. So that's why we did not test N-oxides. However, from a chemical point of view, that is a reasonable option to try that.
Yeah, thank you. Yeah, one more question. So this library of 145 compounds, do you think it's enough in terms of the chemical diversity and the possibility to target almost all possible residues?
So basically, if we consider the opportunities how to develop this approach further, then there are basically two major options. One option, exactly, Yuri, what you're saying, that probably we can go forward to bicyclic compounds as well, for example. That would be interesting, and that would provide us a much more position for further substitution, and that would cover a much larger space, a chemical space. So that would be one clear direction. The other direction, however, would be just, okay, it seems that a high reactivity is needed for these small fragments for the efficient labeling of the protein. However, high reactivity, on the other hand, that is not very much advantageous if we are considering a covalent inhibitor. So the basic difference between the small fragments and the full inhibitor is basically the size in one hand. On the other hand, it is the number of potential non-covalent interactions that could be formed within the binding site. That is the first step of covalent labeling. So therefore, I think that the other direction would be just extending a little bit these small fragments to the real fragment space. I mean, not limiting this approach to the MiniFrag concept, but instead using this pretty large set of warheads on a little bit larger molecules that would provide more options for the non-covalent interactions, and probably we would be able to observe a higher number of hits for the non-methylated and less reactive fragments.
Thank you. Yeah, another question. How do you make sure the molecule optimizations help in the molecular recognition step and not only increase the reactivity of the inhibitor?
So basically, the labeling observed with these covalent MiniFrags, this is driven by the reactivity. So that is very clear. So, however, what we show that there are basically two options. One option that is the so-called ligand-first approach in the developing of TCIs. In this case, one can simply replace one of the aromatic or heteroaromatic ring of the original non-covalent ligand, just making a covalent one or the reactive one. But the difference will be that in the case of acrylamides, for example, you introduce the larger number of atoms as indicated by the four atoms altogether available in the acrylamide warhead, right? That would shift significantly the binding mode of the original compound. However, if you replace like a halogenated electrophilic MiniFrag, then there will be only one single atom addition, and probably you would provide a binding mode closer to the original one. So this is one use. The other one that is more complicated, I mean the electrophile first approach, because as the colleague who asked the question already underlined, in this case, it is purely the reactivity that drives the binding of these very small fragments. And yes, in one hand, as we hypothesized, of course, the binding mode of the small fragment will be fixed. However, just because of the small size of the labeling agent, there might be still some movement altogether with the labeled amino acid side chain and the small labeling. So that makes this type of optimization, I mean the electrophile first optimization, more complicated and more difficult. However, if we are using another approach, not growing, but linking, that would help us a lot, just because we are having the binding modes of both of the fragments. And the question is how exactly we can identify a suitable linker for creating a larger molecule.
Thank you. Yeah, if there was no impact on function, what was the rationale for selecting the initial fragment against MPRO? Was it just a structural screen rather than biochemical, as in the previous example for HDAC?
Okay, so in the case of the MPRO study, we used X-ray screening at XChem diamond light source, and in this case, the basic objective was to identify as many starting points as they can. So they screened, for example, the early covalent fragment library of near London, and also our fragment library. They have also non-covalent libraries as well. So it was an enormous and very international effort. Also, Enamine was participated in these efforts as a part of the Moonlight a project. So therefore, it was not interesting whether or not these small fragments showed any biological activity, but they were confirmed structural hits that might be useful to create a new inhibitors against this, at that time, very, very important protein target. In the case of HDAC, the whole story was different. In this protein, there are basically 8 cysteine residues, and these residues are almost always form a disulfide bond. And in this case, we were absolutely not sure that in addition to the known regulatory sites of HDAC, what is the role and significance of the other cysteine residues? So that's why we first started the project with a biochemical screening, and we focused our attention only to those hits that showed some functional relevance and indicating that that particular cysteines might have some functional role in the regulation of HDAC8 protein.
Yeah, thank you. Yeah, thank you, György. Thank you, everyone who participated in QA session. We don't have any more questions. So again, thank you very much for this really nice talk. Yeah, thank you everyone for joining us today. And please remember that that the library that just been presented by György actually available through Enamine, and that just visit us at enamine.net to find out more about the options to access the library, and not only this library but other fragment libraries as well as classical screening libraries available from Enamine. So thank you very much. With this, I would like to close the webinar, and please just maybe stay for a minute to answer to the survey that you will be proposed at the end of the webinar. Yeah, have a good one.
Bye-bye.